I think we should go ahead and get started. It's a couple minutes after the hour, so hello everyone. I'm Paul. I'm the president, fourth year biotech major. We haven't done introductions since week 1, so I don't know why I did that. But with us today, we have Dr. Eli Borrego, and he is a professor at RIT as of, what, last year?
Yeah.
Have you been here a year or less than a year?
A little over a year.
A little over a year, okay. And he does stuff with plants, specifically plant oxylipins, which are a signaling molecule that plants use to talk to each other. And yeah, go ahead and take it away.
Hi, everybody. Just call me Eli. I actually would like to do introductions because I don't know everybody. So my name's Eli from Texas. The cold is very different. I like racquetball. I like podcasts. I have a cat. His name is Tack. And who are y'all?
I'm Nina. I'm a fourth year biotech major. I'm the marketing chair of this club. I like tennis as well. And I have 4 cats. But they don't have cool science names.
I'm Marissa. Can you guys hear me? My mic can be weird. All right. I'm the vice president of Biotech Club. I'm a 3rd year. I also really like tennis and reading. And I'm also big on podcasts. Have you seen have you listened to In Defensive Plants?
I haven't. I need to. I've heard of it.
They do really cool crossovers with This Podcast Will Kill You.
Okay.
About, like, infectious diseases. So you should look that up. They're really fun.
Nice. I'm going to write that down.
So, hi, I'm Spencer. I'm the secretary, and I am the man responsible for misspelling hormones in the email that told all of you about this. And that was really the high point of the week. And yeah, I do not listen to podcasts much, but I should probably listen to more, so I will probably look into what was just mentioned when I, have free time again in November. Paul, I think it's your cue. Oh, wait, no, you already done. Damn, counting.
I shall go. Hello, I'm Justin Vitus. I am a 2nd year biomedical engineer, currently doing stuff to look for co-ops, but I do have the time to stop in on this meeting, so that's nice. I do enjoy reading, haven't done it for too long, just reading textbooks, but I do also listen to podcasts. One that I recommend is the Magnus Archives, which is this wonderful horror podcast. And yeah, I'm just sitting at home, what I seem to do most days. But yeah.
Hey, I'm Sebastian. I'm a 1st year and biotechnologies also. I'm from Louisiana, so there's that. And then I guess I'll pass it on to Andrew.
Yeah, so I'm a 1st year biochem student, and I was actually just told about this club yesterday. So I'm interested in kind of joining it, and I figured I'd see how this meeting goes, I guess.
Diaz, I nominate you.
I joined a couple minutes late, so I don't exactly know what's going on right now.
Introductions.
Oh yeah, I'm Diaz. Says underneath my name, face. I am a second year BIME, and I don't know what else I'm supposed to say. So hey,
Wait, BIME?
yeah, BIME. It's what the course code calls it.
Oh, gotcha. Okay.
What is BIME?
Biomedical engineer.
Emily, I nominate you.
Um, Hi, I'm Emily. I'm a third year biotech student. I joined the club last year and I've liked all the meetings, so I came to this one.
Kaylee?
Hi, I'm a first year biotech. I came to the first meeting, I just couldn't make it last week. So.
Don't worry, I'm even more embarrassing. I forgot what time it was last week.
Literally no worries. Happy you could make it though. Shafeen?
Hi, I'm Shafeen. I'm a first year BME major. I'm just walking back to class after my chem lab right now, so I'm sorry for the sounds in the background.
Honestly, that's the beauty of Zoom meetings, is that you can be doing literally anything. And Jacob, I guess that just leaves you then.
Can you guys hear me okay? I'm a third year bio major. I am from Greece, New York. And yeah, that's it.
Sweet.
Well, nice to meet you all. This is going to be a very informal presentation, and I want to have maybe several missions. One of them is describing maybe the work I do with hormones and plants. One of them describing hormones in more detail because they're the coolest thing in the world, and also describing this greenhouse that we have connected to Gosnell and how neglected it's been, but that it has a lot of potential for a lot of fun. And I don't know which one, does anybody have which one does anybody want to start off with?
I guess, like, what is a hormone?
Okay. What is a hormone? And you're going to bear with me here because I'm going to use Wikipedia as the screen I'm going to share and talk to you about. So let me share screen 2. Can y'all see that?
Yes.
Okay, so you see, what is a hormone? So plant hormones are a type of signaling molecule. Do you all know what signaling means? Yeah, so it takes information from the world and will use it to modulate in what's called transcriptional reprogramming of the metabolism of the plant or of an organism to allow it to adapt and to be responsive to whatever that stimulus was. So hormones regulate, or signaling molecules is the way the information will travel. And some of the important types of signaling molecules are called hormones. And what separates a hormone from just any old signaling molecule? The hormones require, well, first of all, they're small molecules, small metabolites. So they're, I'd say, under 1,000 molecular weight. They are, they're active in what's called a receptor ligand interaction. So you actually have a hormone receptor and these molecules fit and induce this signaling cascade. that fitting of the receptor to its ligand, that's called perception. So that is how a organism can recognize that a hormone is even there, and it requires a certain threshold to be reached. And a factor of the hormone definition is that these molecules are active in very low concentrations. So there, we can get into some nitty-gritty stuff, but generally they're active in less than nanomolar concentrations. So in plants, well, we can talk about human hormones, which, to be honest, I don't know much, but you know, they regulate a bunch of different processes. They regulate, you know, everything: your blood pressure, your sugar content, your reproduction, your day-night cycle, circadian rhythm. All those processes are under hormone control. And in humans and in mammals, it appears that peptide-derived hormones are the most prevalent. But in plants, we don't seem to have as many peptide-derived hormones, or perhaps they just haven't been identified yet. There's only maybe 3 or 4 that I can think of right now. There's probably going to be more in the near future. But most of the plant hormones, they're, again, these small metabolites. And we can divide them into these different classes, which I will use Wikipedia. And one of the most, well, let me describe the first one that was ever found was ethylene. So ethylene is, you know, the simplest alkene. It's basically just 2 carbons. It's a volatile molecule, so it travels through the air, airborne. It was at one point used as anesthesia, so it can have some type of dampening of pain effects. And this thing was discovered, over 100 years ago by a person. I think he was like a 15-year-old grad student, a PhD student, 15, in Russia. And he noticed that next to his lamp, the plants that were growing, they had this like hooked-like structure, and they would emerge kind of bent. And turns out that it was ethylene being abiotically generated from the burning lamp that was having this effect on the plants. And that's how ethylene was first identified, 100 years ago, or if not more. But does anybody know of a recent molecule that has won the Nobel Prize for being an airborne signal? No? Has anyone heard of that one? NO, nitrous oxide. So that is an airborne signal, and it's, you know, important for a bunch of different processes in cells. And in the Nobel Prize winning speech recognizing it, they say, oh, for identifying the first airborne hormone. And like plant people were really upset because, you know, we've known about ethylene for 100 years, and it's just kind of, again, like this plant blindness that exists in the world. But hormones are important. They control everything in plants. And if you want to be able to manipulate a crop into resisting pests, resisting pathogens, improving yield, drought tolerance, hormones are the way to do it. If you want to engineer a plant to produce secondary metabolites that are important for utilizing as a feedstock into medicine production, hormone biosynthesis and altering the chemistry of hormone production will make that process more efficient. So they can do a bunch of things. So we have ethylene that we described, and there are other hormones, such as gibberellic acid. So gibberellic acid was actually identified in Japan, and gibberellin may be a Japanese word of origin, but it was identified in a fungus called Gibberella fujikori. So what happens in this system, or how this was identified, is a fungus will infect rice, and it will cause the rice to elongate its inner nodes, and it grows really, really tall, but it's really lanky, and it can't support the weight of the panicle of the seeds on top, and it tips over. And this disease was called foolish seedling disease, because it's why is this plant just growing really tall and falling over for no reason? Turns out the reason was that it was infected with gibberella. And this is one of the first examples of fungi manipulating plant hosts through hormones. And that's a very common thing you'll see. A lot of microbes produce plant hormones and are used to manipulate them into better hosts, or used for what's called communication or interactions. And interestingly, a lot of microbes in your gut also produce hormones that are first identified in these plant systems. So like abscisic acid that we'll talk about next, important for defense against abiotic stress. So it's actually a misnomer. Abscisic acid, abscission, is the way that we call when a plant decides to not decapitate, but it controls a limb drop, or it controls a leaf drop. It causes programmed cell death at that junction between an organ that it wants to get rid of and the rest of its body. And that's called abscission. And they thought that abscisic acid regulated that process. So they named it abscisic acid. But it turns out it was really ethylene. What abscisic acid does is it is best known for playing a role in abiotic tolerance. So the most famous being drought. Whenever a plant is under a drought condition, abscisic acid is produced, and abscisic acid will cause the stomata, the opening underneath the leaf, to close. So they call it reduces the aperture of the stomata. And this causes a decrease in gas exchange between the plant and the environment, and the plant can conserve water. So that is the most famous role of this hormone. But in honesty, all the hormones do almost everything, just they have different effects in these different processes. And it depends, and it needs to be species specific, tissue dependent. It's a lot of nuance in understanding the role of hormones. And especially because there's, it's not just humans, it's a bunch of different species that have evolved for their own environments and their own roles. So auxins is probably the most relevant to propagations in plants. Auxin is used to stimulate hormones, to stimulate root production. So the auxin to cytokinin ratio determines what the plant will produce. If there's a lot more auxin, the plant will produce a lot more roots. If there's a lot more cytokinins, it will produce a lot more shoot relief tissue. And auxin and ABA, these again have been found in the human gut, produced by the microbiome. Abscisic acid has recently been found to play a role in diabetes. So I can't remember the direction, but it's interesting that, it shouldn't be unexpected that the hormones produced in your gut by the bacteria in there are going to regulate your processes. And that these communication between bacteria and a eukaryotic host will have shared mechanisms, shared infrastructure. So there may be an overlap between that process and human microbiome interactions with the processes in plant microbiome interactions. Brasinosteroids, these are the only sterile derived molecule or derived hormone in plants that I know of. And there is a lot of interest in this area currently, especially for its role in innate immunity. The brasinosteroid mutant the best understood brasinosteroid mutant was first identified in defensive processes during plant infections with Pseudomonas. And since then, they've realized that what they thought was the brasinosteroid receptor was really a co-receptor for a bunch of other receptors that perceive brasinosteroid, they perceive flagellin, they perceive chitin, they perceive a lot of these other molecules. And we can talk, well, let me mention one other thing about gibberellic acid, because it's going to be a cool experiment, not like a cool, fun experiment that I'm going to do with Emily Wrightstone. So she has a pothos IV, and for all I knew, pothos was completely non-flowering. It turns out that pothos is broken in a gene that makes gibberellic acid. So we're going to spray some GA onto her pothos to see if we can induce flowering, which will be really cool. And yeah, I'm excited about that. Salicylic acid. I am 100% sure you all have perceived salicylic acid, mostly because of its inhibitory content, where it shuts down the enzymes that produce inflammatory responses. So salicylic acid will bind to what's called cyclooxygenase and inhibit it, reducing the production of these molecules and causing inflammation to go away, fever to go away, pain to go away. These classes of molecules in medicine, they're called non-steroidal anti-inflammatory drugs, and they've comprised 60% of the drug market value. So a whole lot of them, a whole lot of money just in one process for drugs. And please feel free to ask me any questions. Like, I love like hearing what people think and like curiosities, because sometimes it makes you consider these things in a whole new world, whole new way.
Yeah, I actually had a quick question. What's pothos?
Pothos, it's an ivy. It's like very common ivy that people have in.
Oh, okay. Is it the one that has been growing here?
Yes.
The one that she brought home. Oh.
She rescued COVID.
Yeah, that thing's looking pretty decrepit. Like anytime it grows an inch, it loses a leaf.
Is this the hanging plant?
Yeah. It's kind of sad. I feel so bad for it. If I had like one more ounce of motivation in my body, I would go and get it and just show it off. But it's very unruly.
Motivation is a premium resource at this point in the semester.
They usually enjoy being in indirect sun, but because of that, they want to grow really long.
Yeah, so, it's in a very shady spot right now, so we might move it.
Strigolactones, these are a molecule group that is produced by produced by plants, but first identified in parasitic plants. So you know how we usually think of plants as producing, being autotrophs and producing their own food. Like, that's one of the cool things about plants. They can turn sugar. They can make sugar from air. So that is one thing, though, that these parasitic plants can't do, and they have to feed off of other plants. So they form structures that are more like fungal, parasitic interfaces, and they can pathogenize plants. Some plants can even pathogenize fungi. They're called microheterotrophs. But the strigolactones were first identified in Striga. And I kind of like this story because Striga, I think, is a Slavic word that means witch. And this plant is called witchweed. So kind of cool. But yeah, they find it in a bunch of different processes with plant interactions, plant fungal interactions. And there is other molecules that are least understood, and actually this is where I come to learn about, oh, there's new plant peptides. So every now and again I'll come and check this website, and that's a very fast way of identifying them. Oh, here's a good one. This one, Caricans. This is it's a word from one of the Australian Aboriginal languages that means fire, or it means smoke. And it's actually produced from the burning of plant material. And it's a molecule that induces germination of seeds and other processes. They've started to studying it with its role in pathogen resistance and insect resistance. And it's kind of like, it has a certain place in my heart because I saw Caracans, the first story of it emerge. And, you know, when I was in grad school and I started paying attention to scientific literature. So every now and again, I'll see another big story about Caracans, and then I'm like, oh yeah, I remember that. Yeah, but there's others. And what I want to emphasize is, I haven't talked about JA yet, but what I want to emphasize is that the majority of these molecules are or exist in classes. So there's a few molecules that look like ABA. Some of them are storage forms. Usually they're conjugated with amino acids that are storage forms. Auxins, there's a few molecules that look like auxins. There's a few steroids, a few cytokinins. There's just ethylene. Gibberellics, there's a few. There's a few salicylic acids, a few strigolactones. Some of them don't have direct biological function, but they are modifications, or other species may perceive different versions of them. But they're all generally structurally very, very similar. JA belongs to a group of molecules called oxalipins. And these are what I think about day and night. These molecules are derived from fatty acids. Have any of you ever heard of omega-3s, omega-6s? The diet tells you consume omega-3s more than omega-6s. Well, that is because they turn into what are known as oxalipins, these oxidized fatty acids that look extremely different from each other. They're alcohols, aldehydes, esters, dicarboxylic acids. One of them are called green leaf volatiles. So when you mow the lawn, that odor of fresh cut grass, that's a warning signal that plants use to communicate with each other, and for plants to communicate with other parts of itself through the air. And all these molecules in humans have revolutionized medicines. There are several drugs that are oxalipins, or usually methylated oxalipins, to make the bioactivity a lot increased. And these molecules, they come from fatty acids. All of life, anything that has a cell membrane is going to have fatty acids. And that's a common universal thing of these oxalipins. They're found everywhere. And for the majority of their history, they were ignored because they were thought to be oxidative mistakes. Like when you're extracting lipids, you're going to have oxygen get in there, and you're going to have these oxidation processes occurring. And people for the longest time assumed, okay, these are there, we're seeing them, but it's just because we suck at extractions. But no, in reality, These molecules are enzymatically generated. They have regional stereospecificity that you change one version of S to an R, or you change a double bond from cis to trans, it no longer fits in its normal receptor. It fits into a different receptor, and it could potentially have opposite roles in whatever process. And most of that is well understood in humans. Like, that's the reason it's revolutionized medicine. The reason the NSAIDs are so important and that so much money is generated to developing new ones and ones that are more tissue specific is because they shut down these cyclooxygenases that create these oxalipins in humans. Plants, these exist. We have a very poor understanding of what they do, and that's what our lab's trying to study. They seem to play a role in the sense that they have broad activity as they do in humans. They govern what seems to be almost every process that they've been explored in. So I mentioned that abscisic acid will close its stomata. So this is just one example of what can happen with oxalipin signaling. So the stomata closes also in a defense against bacteria. So a plant will perceive the flagellin or perceive some external decorations on a bacterial molecule, on a bacterial cell, and close the stomata so that the bacteria can't get into the plant. Bacteria have the ability to inject things into a plant cell. And one of these things is called chronotin. And chronotin looks very, very similar to JA, to jasmonic acid, except it binds to its receptor 1,000 times better. And when JA signaling is engaged in guard cells, they open and the bacteria can swim through. So this is one example of how an organism can manipulate another organism using oxalipins and hijacking their oxalipin infrastructure, their oxalipin machinery. Does anybody like hot peppers? Yeah. Okay. Y'all know what makes it hot? Capsaicin, yes. So capsaicin is perceived by mammals and mostly is hypothesized to have evolved because when mammals consume seeds of these capsaicin plants, they're related to tomatoes and potatoes. They're solanaceous. So when mammals consume the seeds are degraded. And yet when birds consume the seeds, they're able to pass through the digestive tract and have increased germination rates. But the plant is, evolution's a very funny thing where this molecule was generated that only targets a mammalian receptor called a capsaicinoid receptor. And the ortholog in birds is unaffected. So birds will eat capsaicin and peppers without perceiving the heat. But mammals perceive the pungency and then try to avoid it. Unless we're like humans, we might want to eat more of it. So these capsaicinoid receptors, they're involved in, like we know they're involved in pain. There's some medicine that's actually like spreading capsaicin on your joints because it's got an whatever the word is, analgesic effect. But you don't make capsaicin, so why do you even have capsaicinoid receptors?
Aren't they like thermal receptors? Isn't that the thing?
They will signal like it's perceiving heat, and they're what's called a ligand-gated ion channel, and that means that there's a little there's a small molecule that will go and attach to them and trigger their effect. So in humans, well, in animals, that endogenous ligand are oxalipins. So there's some commonalities between capsaicin and pain that can be explained by the oxalipin's role in regulating those processes. And has anybody ever heard of cannabinoids? I see smiles everywhere. So cannabinoids function, well, at least phytocannabinoids function by targeting cannabinoid receptors. So there's cannabinoid receptors in the brain that perceive these phytocannabinoids and may induce a psychoactive property. There's cannabinoid receptors in the gut or in muscles that perceive cannabinoids that may induce other relaxing properties or digestive enhancing properties. And we don't produce phytocannabinoids, but we do produce endocannabinoids. And the endocannabinoids are oxalipins. And these two examples are my favorite examples of how plants, in this case, are able to hijack mammalian oxalipin signaling to either, well, in both cases, probably to deter feeding, but now we've kind of enjoyed them. And that is the reason, like we take them because of their hijacking of our systems. So that's what I study in my lab, and because of these molecules existing as hormones and a concept. Have y'all ever heard of signal crosstalk? More or less, where one signal can influence another signal. There's additive effects, antagonistic, synergistic, negative. A lot of the crosstalk is also explored in what's called hormone crosstalk, how one hormone can influence the effects of others. And one of the most famous examples, again, because of oxalipins, is a balance between between the model oxalipin called jasmonic acid, named for its plant of origin, jasmine. So the smell of jasmine is a derivative of jasmonic acid. Fun fact, there is a human pheromone receptor, and this human pheromone receptor, the best thing that binds to it is a plant oxalipin. So it's actually sold in colognes and perfumes that are used, but it's also what's called a jasmonic acid, and this JA is antagonized by salicylic acid. So salicylic acid shuts down cyclooxygenase in humans. That's why it's got a large market value, blah, blah, blah. That same balance exists as paralleled in plants, where salicylic acid can shut down jasmonic acid, biosynthesis and signaling. And jasmonic acid can shut down salicylic acid signaling. And salicylic acid definitely has a greater effect than JA against SA. But here's the thing, so that's negative crosstalk. When those systems are exposed to ethylene, SA can no longer shut down JA. So it's a conditional response. And from what I've studied, from what I've seen in the papers I read about this, is that once ethylene is induced, the cell no longer uses SA to shut down JA. So there's all these really cool, again, nuances. Another example is JA's role in fertility. In nearly all plants that are studied, JA controls male fertility. Without JA, the anther usually doesn't form, it usually doesn't dehisce, so it doesn't open up and let the pollen come out. Sometimes the pollen's viable, it's just like locked up. J deficiency in maize is even more extreme because, so while rice has the panicle that contains the male and female flowers, the male plant, or the maize plant, is completely unusual compared to all other plants in the world. The male organ is the tassel. It's on top. The flowers there are completely male. The flowers of the ear or of the cob are completely female. The process that regulates that is jasmonic acid. Jasmonic acid is almost like a testosterone for maize. This male fertility is exaggerated. The role of it in male fertility is exaggerated because the male organ becomes feminized. It produces female flowers and it can produce actual seeds that will be viable. The ears, without jasmonic acid, grow gigantic. It almost like hyper feminizes. Problem with JA deficiency, you think, oh, wow, this is awesome. We can use it to improve yield, to improve food. You know, these plants are super, super susceptible to soil-borne pathogens. You plant them in the field, they're dead in 3 days. You have to grow them in sterile soil because of the role of JA in defense. And where was I going with this? Yeah, that is what we study. And because of the role of hormones on each other, I do a lot of analytical chemistry. So I actually measure the molecules with LCMS. I study processes with genetic evidence. So we make mutants. We have lines that can no longer make certain oxalipins, and we study how they've changed in a certain phenotype relative to their wild type, and then try to complement them with the molecule that they're missing to provide good evidence that that's the role of that molecule. And you know, exploring all these hormones is going to hopefully change how we approach agriculture and provide a similar revolution as we've seen in medicine. And this kind of segues us into the next part of propagation. So we have this greenhouse. It is right now a little rough looking, but it's got a lot of potential. And what we were able to do is get a little bit of money from the department so that we could buy pots, we could buy soil, and we can help make it more attractive for, you know, recruitment purposes, for just, you know, mental health, enjoying the plants. And I enjoy them. I go down there whenever I'm stressed out. That is a lot, just to go talk to them. Can y'all see this? Yeah, awesome. I wonder if it will just show up if I do that. Yeah, okay. Can y'all still see the big screen? Cool. So the week of spring break, I spent it in the greenhouse. I was avoiding writing papers, writing grants, and I cleaned up a little bit of it. I arranged everything phylogenetically. So you have all the cacti, you have the agaves, the aloes, you have the euphorbs, kind of in this area. And so you can see it was a little bit rough. It has gone a little bit downhill from that because of COVID, and we weren't here for 20 weeks. This is the other side. There's some orchids, some more euphorbs, some cycads. So there's a lot of a diversity within this greenhouse. Down here you have some pitcher plants, and in here there's a Venus flytrap. Does anybody know where Venus flytraps are native to?
Carolinas?
Yeah, good job. Yeah, we always think like they're they're from this like more exotic stereotyped place, but yeah, they're from the Carolinas. Now here's a pitcher plant starting to form, a pitcher starting to form on the plant. Some very unusual plants that reproduce by little flowers inside brachies, which is really cool. Here is one of the, what's the best word to put this? Here's one of the ways the plants have been treated currently. So these have existed for a while, and I'm not sure who's been able to take care of them, but there's this condition called root bound. So the plant basically takes over the entire pot. It can no longer grow. And here, this particular agave is like making a whole bunch of little babies, and they've just conquered this pot. But what we can do is transplant them and create, out of this plant, I think I got 12 pots. Here's a before of what's called mother-in-law's tongue, because it's long and sharp. And if you put yourself, like, it'll cut you. It's usually very pokey at the end. But yeah, they're completely overrunning their pots. And here's one I did today, actually. And this was an agave that was overrunning its pot. And then what's interesting is these leaves, I never knew this happened before. These leaves, they dry out, but before they, or as they're drying, they transform the midrib, the central vein of the leaf, into a root. And it only happens on the underside of the little stem. So there must be some gravity effects that allows some hormone-mediated reprogramming to occur. So that was really cool. I'm sure it's been reported. If not, that's neat. That could be a little paper. But here's an example. This is a picture from March. You can see this, that's that same species of agave, kind of like overrunning its pot. It knows it's root bound, so it's trying to send its babies outside to go and conquer some other land. But it can't, because there's just air. So I transplanted them. Actually, these were today, and they will look a lot better. I give them maybe a few months to clean up. This is the mother-in-law's tongue, also known as Sansevieria. And here I transplanted it into 3 pots because I'm very afraid of breaking it up into individual plants, because they're too root bound and they're too interwoven with the roots that I feel that if they're broken up anymore, they'll be damaged. So once they start, making new roots and starting to colonize this pot better, then they can be broken up. And then here's what it's kind of looking like. So a bunch of little pots, and the plants, these are all succulents. I've noticed that at RIT, succulents are very popular plant to have. And part of me wanting to be here to share this with y'all is I'd like to kind of get like a plant propagation club going, where whoever's interested can come and propagate plants. We could sell them once a year to freshmen, $5 a plant. That's a pretty good deal. And then use the money to buy soil and buy other exotic plants. Kind of just like a self-perpetuating cycle, if anybody is interested. But yeah, that's the end of the story. Does anybody have questions or is interested?
I personally know I'm very interested.
So also, about once every other week, we have a plant science journal club where we meet and discuss high impact paper in the plant sciences. And so far it's been going pretty good. Yeah, Justin, Paul, Nina, Jacob. Yeah. So any questions about hormones, oxalipins, plants?
Yeah, do you think that we could make the propagation process happen faster if we just sprinkle a little auxin on it and promote root growth?
That can definitely happen. I purchased some auxin. It's going to be here in a week. I think it's better for plants that are harder to propagate. So like cacti and agaves and aloes, those will be making roots very easily. Other plants that are maybe more woody or semi-woody would definitely need auxin to help. So while I'm mainly focused on the succulents, we also have the other side of the greenhouse has all the orchids. And I was able to transfer a few of the orchids into new pots. I gave them clean soil back in March, and they bloomed for the first time in a long time this summer. So I think there's a lot of potential for a little bit of care to go. And this particular section of the greenhouse it's what's designated as the demonstrational greenhouse. So it's kind of for learning and interacting with all these different plants.
How are you propagating the plants? Because I know you can do it like different ways.
So there's two overarching ways of doing it. So we got reproductive propagation and then there's vegetative propagation. So these are all vegetatively propagated. The plants usually send off shoots or send off pups that we can move into new soil. And at this stage, most of them are already making roots. They're trying to get out of their pots. They just need some help to get them into a new pot. Some things like cacti, like you can't see it now, but there's these really tall cacti that are actually flowering because I fertilize them. And I'm excited, but once they drop flowers, I'm gonna chop them up and I'm gonna well, at least into like more manageable pieces and then propagate them so that they're not gonna fall over and damage themselves and they'll be able to be, amplified. So yeah.
Do you have a meeting time for this club yet?
I do not. I'm still trying to gather people that are interested, and it could be, with the occupancy limitations, we may have like people 2 or 3 at once in the greenhouse area, and yeah, I haven't been able to establish anything yet. So far, just gathering people that are interested.
If we are interested, how would we get involved in that?
Well, I suggest just email me and I will put together a collection of emails and send out a message for when a good time is to meet. Yeah, I think that's the best way to approach it. And I just typed in my email address down there.
And I'll have Lop send it out in the weekly email as well.
Cool. And if anybody's interested in doing research on oxalipins, just let me know.
It's pretty cool research.
So a disclaimer, Nina, Paul, and Jacob are all doing research with me.
If anyone has any tips on germinating Arabidopsis, I am all ears because it's being very fickle.
Nope. I definitely have gained a newfound appreciation to the smell of freshly mowed lawns, because it's really just a bunch of grass screaming and crying for help. It's macabre.
Wait, but is there like a reason for them to do that? Like, even if they scream and cry for help, like they couldn't do anything. So
Oh,
why,
Eli,
why,
Eli,
why do they do it? I don't understand.
Do anybody want to answer?
I believe the system originally evolved to whenever the plants were damaged, like insect biting. So if an insect was eating it, then it would release these signaling compounds to then have, I guess, make the plant taste bad for the bugs. Then it would stop eating it, like an anti-herbivory response to it.
Also, I think predators of those bugs, it attracts them. So like I think parasitic wasps is one of them, and also like general like plant defenses, like the cell walls maybe increase like deposition so it becomes more strong. But cool.
And one of the things these green leaf volatiles can do is a phenomenon known as priming. So Paul and Nina, perfect answers. And one of that particular system is called tritrophic interactions, because there's three levels of the food web. And this idea of priming is more of internal signaling. So these green leaf volatiles are emitted, and plants respond to them initially in something called induction. But plants that are exposed to green leaf volatiles, if they are within 4 days exposed to an insect or to a different stress, the stress responses will skyrocket a lot earlier and to greater extent than of a plant not have been previously exposed to green leaf volatiles. And you can kind of almost think of this like a vaccine, but what people are finding is that we don't really know how it works yet, but the hypothesis that best explains this is what's called RNA-dependent DNA methylation. So parts of the genome, of the plant genome, are methylated or unmethylated to allow accessibility by transcription factors to those areas that are responsible for allowing the response, the induction response, to occur faster. So that's still an area that's highly explored and that we'd be interested in, because I have a corn line that can no longer make those smells. So kind of cool.
Sweet.
Thank you all for coming, and I'm glad I met you, and thank you for listening.
Yeah, it kind of wrapped up at right around the perfect time. Next week, as well as Plant Science Journal Club, we meet via Zoom every other Wednesday, and then this upcoming Wednesday is the one that's like not this tomorrow, but next Wednesday. So I'm sure you can send Eli an email about that as well, and he'll put you on the mailing list. So
who's presenting?
Hannah said she'd present. I should remind her.
Yeah, because I haven't seen the paper yet, like go up.
Okay.
Cool. All right. Well, thank you guys so much for coming. Yeah, and sweet. Have a great. Oh, I guess a few other things. Apple picking is tentatively scheduled for Sunday, October 4th. We'll be sending out a sign-out sheet for that. We just need to get approval from clubs, which is taking a ridiculously long time. Also, next week, we will have Dr. Gary Skuse be giving a talk about biotech that doesn't have to happen at the web bench. So if you're not able to make it, we'll record that one and post it on our YouTube channel. And if you guys have any questions, just shoot us an email. Sweet. Thanks, guys. And thank you, Eli, for presenting.
Thank you, Eli.
Thanks, Eli.
Hope you weren't too quiet.
Bellissimo.
We're doing Biotech Club lingering. Is the e-board lingering?
Yes. Yes.
Right. Paul, first,
Bruh.